Welcome to the MCAT Basics Podcast, brought to you by the physicians at Med School Coach. Each week, Sam Smith breaks down high-yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to a bonus episode of Sam's MCAT Basics. This bonus episode will cover cell differentiation mechanisms, and this is really in terms of development. So a little bit ago, I did a podcast on development in the reproductive system. And in that podcast, I went over spermatogenesis, oogenesis, and then I got a little bit into development. And all I talked about in terms of development was how the blastula undergoes gastrulation, and you have this invagination that forms. And from that, you have the germ layers form. And then you have neurulation, and etc., right? You have all these stages that kind of are defined and we know occur during development. What I didn't talk about was how do the cells that are all one type eventually become lung cells, heart cells, liver cells, all these different cells, right? And they're all very distinct from one another. So how does this process occur where you have this essentially blank stem cell become one of these other cell types, and how does that process work? And so what I want to introduce is a few different mechanisms that stem cells use in order to differentiate into different types of cells. All right, so real quickly before I get into cell differentiation mechanisms, let's put this back in perspective of where we're at in terms of reproduction. So to start, obviously, a female has been inseminated. You can guess how that happens. And then next, sperm motility actually increases in about an hour or two of just kind of hanging out inside the female reproductive tract. That's called capacitation. Once these sperm gain full motility, then they swim up to the fallopian tube where they go and they bind to an egg. And the sperm specifically bind to what's called the corona radiata. And then what happens is you have all these sperm binding to the corona radiata, and sperm are kind of wiggling their way in, and they continue to move inwards towards the egg. And eventually they reach the next layer called the Zona Pellucida. And when a sperm binds to the Zona Pellucida, the acrosome organelle on the sperm bursts. And if you remember from the development podcast, the acrosome is just this organelle that's on top of the sperm, kind of looks like a helmet, and contains all these digestive enzymes that help the sperm get into the egg. So at this point, a single sperm hopefully has broken through the Zona Pellucida and is entering the egg. And at this point, the cortical reaction occurs, which essentially goes to lock all the other sperm out. So you have this locking out of all the other sperm. And at this point, the egg is a secondary oocyte, and it undergoes its second meiotic division. And the sperm nucleus then fuses with the ovum. Both of these are haploid before that, and then once they fuse, they form a diploid zygote. That diploid zygote undergoes blastulation to form the blastocyst. And then you have gastrulation, which is just an invagination of the blastocyst, which the germ layers form from this invaginated blastocyst. After that, you have neurulation. So that's kind of where the last podcast left off. So now that you kind of are back into this zone of mind, let's get into how these cells differentiate. And it's important to note here that cell differentiation is occurring throughout all the development steps that I just listed off. So during that whole time, you have cell differentiation. So cell differentiation is brought about by both intracellular factors and extracellular factors. So I'm going to go over both of these. But before I go over those, it's important to understand how different cell types are defined. Right? What exactly makes a heart cell different from a lung cell? If you took that out and you looked under a microscope, would you really be able to tell which one's a heart cell and which one's a lung cell? Probably not. And that might not be the best example, obviously, because heart cells do look a little bit different, I think, in under the microscope. But say you took, a normal muscle cell and you looked at a heart cell. Well, would you really be able to tell the difference? Probably not. So cell types are actually defined by their gene expression profiles. So essentially, you can determine a gene expression profile, in the lab at least, by sequencing all the transcripts, and this is called the transcriptome. So you look at all the mRNA transcripts that are being produced by this cell, and from that you can say, okay, well, this is producing a bunch of mRNA transcripts associated with heart cells. We know this is some kind of heart cell. For instance, heart cells express this very specific kind of myosin binding protein called myosin binding protein C, where C stands for cardiac. And so if you were to look at all these cells, let's just say, you took you took tweezers and you pulled one heart cell, you pulled a lung cell, you pulled a face cell, you pulled a tongue cell, you pulled an eye cell, and you just line them up all in front of you. Well, one way to say, okay, this is a heart cell is to look and see, can I find that very specific transcript for that myosin binding factor or that myosin binding protein? And so this all goes to say that you can define cell types based on their gene expression patterns. And that is actually how, at least in the science that I know, that is how scientists do it. They define different cell types based on their gene expression patterns. All right, so what is our blank canvas for development? Well, these are our embryonic stem cells. They can become many different types of cells. They are kind of our starting material for the body. And quick Game of Thrones reference, these embryonic stem cells are kind of like the many-faced god, right? It can become anything, it can take on any face, it can take on any type of cell. Since cell types are defined by their expression patterns, then what must determine the different types of cells is some mechanism that can turn off genes and turn on specific genes, right? Because every single cell has the full genome repertoire, right? They have the gene for every single protein that can be expressed in every single cell in your body. So in order to control these cells and turn them into the cells that the body wants, it has to turn off and on different cells at certain patterns, and that goes on to then determine the gene expression patterns. So at this point, I hope your head's starting to kind of mull around with things that could turn on and off genes. One of those is obviously epigenetics, and I'll talk about that a little bit. That's one of the mechanisms. But let's get into those mechanisms. They're either intracellular mechanisms or they are extracellular mechanisms. All right, let's talk about intracellular mechanisms first here. So cells rely on intracellular signals or cues to tell them what kind of cell to differentiate into. The very first mechanism I want to present is called asymmetric segregation of cellular determinants, which is a mouthful and is not quite as convoluted or complex as that sounds. So all that's saying is that an unequal distribution of these molecules that affect transcription can play a role in determining what type of cell the stem cell differentiates into. So, for instance, imagine a cell and it's full of these different transcription factors, right? What are transcription factors? Well, transcription factors are just these proteins that bind DNA and turn genes on. So they activate genes. So let's say you have this cell, half of the cell is filled with one type of transcription factor. The other half of the cell doesn't have this certain transcription factor. Well, what happens when that cell divides? Well, let's say you split that cell in half and now it divides. Well, one of the cells that it divided into is going to contain all these certain transcription factors that were in that cell, whereas the other cell isn't going to have any of those certain transcription factors. So that's just asymmetric segregation, right? You have specific transcription factors segregate to different parts of the cell, and then when the cell splits, all these transcription factors go into one of the daughter cells instead of the other. And then in that way, you activate these different genes. So quick example here. Let's say we have an embryonic stem cell, and we are looking at this transcription factor called insulin-producing factor 1. And so as you could guess, this actually stimulates the cell to produce insulin. Let's just say this is completely made up, but pretend it's real. And so you have this insulin-producing factor 1 all segregated to the bottom side of some cell. Now, when that cell splits, one of the daughter cells gets all of that insulin-producing factor, and the other cell doesn't. Well, now this cell that gets all that insulin-producing factor actually may go on to then become a liver cell. Obviously, you're going to have a bunch of different transcription factors that are specific, but just taking the example of one of these, you can see that you start to get these characteristics of certain cell types by this asymmetric segregation of cellular determinants. And so what other types of molecules can be segregated during this mechanism? Obviously, I talked about transcription factors, but there's also epigenetic factors that play a role in this segregation. You can have histone deacetylases or histone acetylases or proteins that methylate DNA. You can have all of these different epigenetic acting molecules also segregate differently, and those can play a role in, silencing genes or turning on genes depending upon where they are located within each of these daughter cells. And then on top of these epigenetic factors, you can also have these other signaling molecules. For instance, I read a paper in which there was a signaling molecule called chondroitin sulfate that actually acts as a signaling molecule. This can segregate asymmetrically and then result in cells differentiating into different types of cells. All right, and so that's kind of the main internal differentiation mechanism, is that asymmetric segregation. And I hope that this kind of helped you see in your head what's going on with all of these transcription factors or epigenetic factors moving to a certain side of the cell and then that cell dividing. If not, go Google a picture, and it shouldn't be too hard to see. All right, let's get into the extracellular signals that tell cells which type of cell to differentiate into. The first and most important extracellular cue to understand, or signal to understand in terms of telling cells which cell to differentiate into, is called induction. And this is when one group of cells that neighbors another group of cells induces that group of cells to differentiate. And this can happen in a few different ways. This goes back to cell-to-cell signaling. This can happen in a direct cell-to-cell binding, and this is called juxtacrine signaling. This can happen in a release of some kind of molecule that diffuses from one cell that binds to some extracellular receptor on another cell. That's called paracrine signaling. And what happens then is that these signaling molecules, or this direct cell-to-cell binding, induces a signal transduction pathway in the affected cell, and eventually that signal transduction pathway results in some kind of difference in gene regulation. And as I said before, the end result has to be that gene expression is changed to a different pattern, right? Because that's what defines a cell. Induction sort of reminds me of what happens when you spend too much time around a friend. Eventually, you start picking up a little bit of their mannerisms here and there. At first it might be something that they say. Eventually, it might be something that they do. And before you know it, you are basically your friend. The second type of extracellular signal that I want to talk about is mechanical signals. So interestingly, it's recently been shown that different mechanical signals can cause cells to differentiate into different types of cells. So for instance, one study showed that altering hydrostatic pressure within a solution containing stem cells actually changed the type of cells that were eventually produced by these stem cells. So, at one pressure you produce heart cells. At one pressure, you produced lung cells. At one pressure, you produced eye cells. Whatever. It worked sort of like that. Obviously, I'm definitely simplifying it, but that's kind of interesting, right? And they also showed that mechanical stress, so putting tension on these cells, could actually cause the same kind of effect. And the most interesting part of this is that this change in differentiation was actually observed in the absence of other signaling. So they took away other signaling molecules that would tell this cell to differentiate into whatever type of cell it was going to. They took away all those signals and instead just used mechanical signals, and they were still able to produce the same differentiation results, which is very interesting. It suggests that these mechanical signals actually play a big role in determining the fate of these stem cells. All right, so that is a brief introduction into different cell differentiation and mechanisms. And so I introduced internal asymmetric distribution of transcription factors, proteins, epigenetic factors, signaling molecules. Those all affect differentiation. Then I introduced external signaling. And so this can be done by adjacent cells, which is either juxtacrine or paracrine signaling, or it can be done by mechanical forces. So applying pressure, applying torsion can produce different kind of progenitor cells. And in the end, I think you can take kind of a holistic look and say, okay, all these forces are working together to determine what type of cells are being produced from a stem cell. So you're having different distribution of transcription factors. You also are having modified mechanical forces, while at the same time, different cells around this cell are actually kind of pushing it towards a certain cell type. So it's very complex, very convoluted, but I hope this helps give you a little bit of a picture into how stem cells differentiate into all the different kinds of cells of the body.
Each episode of MCAT Basics is brought to you by Med School Coach. To access Med School Coach services, including MCAT tutoring and medical school admissions advising, visit our website at medschoolcoach.com. Good luck as you prepare for the MCAT, and we hope you tune in again next time.
Come on, Murph. I'm trying to podcast over here, dude. Sam's MCAT Basics is written, produced, and recorded by Murph the dog. Special thanks to Murph the dog for this episode.